All right, thanks, David, and thanks also to the GETA administration, Danielle and Danny and Chris. I've known them for a long time, and obviously Sarah and her husband. Andrew. Oh, yeah, Andrew. I never refer to him by his first name. He's terrific. As David alluded to, have a career researcher, and I had a lifelong interest in understanding disease mechanisms. How do changes, whether it's an acquired injury or they're a genetic injury, how does that lead to the manifestation of a disorder? And with that understanding, there's always the hope that, well, if you understand something well, you really understand how to intervene. And that's always been a big part of my whole research journey. And where it's led to, I've been at I did my PhD work at Melbourne University, went to the University of Massachusetts where I did some further studies, and came back to Melbourne, and I've been at the Florey since about 2000. I was the director of the Florey from 2018 till about a month ago. I stood down from that position in order to work with a company called Praxis. I co-founded that in 2015 and have been involved in there. And the whole point of Praxis was to take the latest advances in genetics, the latest advances in biology, and to try and deliver therapies. Because for me, it's great to understand diseases, it's great to understand how you might go about treating them, but if you actually really want to deliver something, you really have to roll your sleeves up and get involved in the actual delivery of it. And that means working with commercial organisations, that means raising hundreds of millions of dollars to be able to do that. That means developing teams. Praxis itself is located in Boston, in America. It's got 140 people now. It's got an incredible executive team, the CEO, and executive leadership there have delivered multiple drugs onto the market, so they know how to take things from an idea into actual delivery of a product. And as you'll see today, I'm going to talk about the things at Praxis that are near and dear to many of the people in this room, things that are very near and dear to me, and things that I'm passionate about trying to get over the line. Because it's a company, we have to give these disclosures about forward-looking statements. It's up there and it's going to be in the presentation. So I ask anyone who needs to look at that. So I'll talk a little bit about background, and this is actually a slide I borrowed from Sam Berkovic, who Danielle mentioned earlier, and it really looks at the changing face of epilepsy. And this was epilepsy in the 40s, and this is where modern day, how we got to where we are today in actually understanding the genetic basis of the disorders that many of the people in this family and this group of families is facing with their own children. And it started with twins. And these were, there's a famous epileptologist, William Lennox from the US, who was studying twins and asking the question, does epilepsy occur, if you're an identical twin, what's the chance that twin one has epilepsy, what's the chance that twin two will also have epilepsy? And that rate of concordance was much higher in identical twins than non-identical twins. So that immediately, because with identical twins, you're genetically pretty much identical. So that tells us that there's something genetic that's coming along with it. And that was the really first signs that epilepsy was a gene disorder, not some disorder that was acquired through an environmental toxin or an injury, which can occur. But far and away, most epilepsy, whether it's the rare epilepsies that today we're talking about, or even the very common forms of epilepsy, there's a huge genetic component. And he studied many, many twins. This is one, Caroline and Eleanor. They both had early morning tonic-clonic seizures and myoclonus. Caroline at 16 years of age, Eleanor at 17. And these girls also went on to develop psychosis. And you see almost similar age of presentation there. EEGs were very similar. Something they shared in common was a genetic change that resulted in their epilepsy. So it was a big leap from there to where we are today. And as Danielle said, that leap, that change from twins to actually an understanding of this gene and this disorder happened in Australia, happened as a result of the pioneering work that Sam Berkovic and then later Ingrid Scheffer, as a dynamic duo, I call them, really defined that area. It's now taken, obviously, people around the whole world have cottoned onto that, and the work is expanding as it should in science, but they really pave the way for that understanding of first in common, and now the unequivocal understanding we have in rare diseases. When you look at epilepsy, if you're going to try to intervene in a disorder, many disorders have got compelling genetics. But what is it about, what distinguishes epilepsy above them as an area that we think has got the greatest early chance of having deliverable therapies? And if you look at it, one of the databases that's available in the literature is called Online Mendelian Inheritance in Man, OMIM. And that's just a database of articles and research articles that are investigating the genetics of a disorder. And if you just look there and you look at epilepsy or seizures within that database, and that's the two red blocks, you can see across all the other disorders, those two conditions, epilepsy or seizures, is far and away the most common linkage to a genetic study than anything else put together. So that tells us that there's a huge genetic signal. And this is just the field. It's not a deliberate thing. It's just people are doing studies and they're finding these linkages, and then these things get picked up by this database, and you can see a massive overrepresentation. So I think that tells us that of all the neurological disorders, there's a very strong and clear link between our genes and the disorder in epilepsy. And that's a really important link to understand on the journey, not only to diagnosis, but to therapy. And what else it's doing, as we know more and more, and people here might see their gene that affects them and their family in this group, is that we're starting to see classes of genes emerging. Again, this was from a nice study led by Sam Berkovic. It was summarising current thinking. And you can see here ion channels, genes involved in synapses, genes involved in structural rearrangements, the so-called mTOR pathway, genes that are involved in the regulation of DNA. And you can see many of the genes that are mutated that give rise to common and rare diseases are starting to fall in these clusters of classes of genes. And that also gives us an idea of how to think about it, how to think about important issues like how you go about making a drug against that, whether it's a gene therapy or a small molecule, how reversible the effect, and David touched on that. Treating early is important because more and more things happen that make it harder and harder to reverse the disorder. We don't know where those lines are for various disorders. Maybe for a structural gene that changes the structure of the brain, it might be harder to reverse the effects of that than something that doesn't. Understanding these things tells us a lot. It creates imperatives. It really motivates us to think about how we go about delivering therapies. If we think about how genetics works, and this is sort of a blank canvas framework for any genetic disorder, and then there's two really major concepts. You say, we all know about genes, and there are individual mutations in genes, and that bottom axis pretty much says how common are those genetic changes in the population, how often do you see them, and the other thing is how strong is that effect of that gene variation. Now these two axes help you map the disorder, and it gives you a lot of clues as to the mechanisms that produce the disorder. Now for epilepsy, we first saw it studies in twins, studies in families that led to the identification of genes and giving us unequivocal evidence of a link between a gene and a phenotype. And then more recently, the advent of next generation sequencing that allowed us to completely sequence a patient, pretty much all the important genes and the important parts of genes that help us understand why a disease occurred, allowed us to map. And what we are seeing is that epilepsy is falling on these axes. There's rare disorders with large effect sizes, and pretty much that's why we're here today. Single gene disorder, mutation that occurs, ultra rare mutation occurs and gives rise to a disorder in the child. This gene is not represented in the parents. It's a de novo mutation by and large, and it causes a very severe disorder. The second part of this is really going back to the more common forms of epilepsy, where rather than one single gene having a massive effect, it seems as though it's the accumulation of small genetic changes that are common to all of us, We've all got these different genetic changes that determine our height, colour of our eyes, whether we've got curly hair or straight hair, all of these genetic features. But if you accumulate a particular set of those changes, you increase your chances for epilepsy. And that's really the second wave, I think, of precision medicine in epilepsy is probably going to occur in box 2. Where we are now, and where a lot of attention is going, what I'm going to talk about mostly today is box 1. Rare diseases, and each gene has got a very, very strong effect. And so we know that mutation caused that disorder in that child. We have to do something specifically about that single gene mutation. And now we sort of understand from twins, we've gone to this really, this is another slide I borrowed from Sam Berkovic, a slide that talks about the spectrum of epilepsy. If you look at prevalence, how common it is, and you look from rare monogenic diseases on the left, which I said is what we're sort of going to talk about today, to the fully acquired epilepsies. A lot of brain injuries, whether it's a cardiovascular or a tumour or a head injury, can give rise to epilepsy. And we see that, unfortunately, in the military with traumatic brain injury caused by a lot of blast injury, can give rise to epilepsy, purely acquired. On the left, purely genetic. And then in the middle, things that are a bit mixed. And everyone's going to have a various mixture of some genetic component and some environmental component. Understanding where that is, understanding which genes are susceptible to environmental influences, etc., again, is important in diagnosis, is important in therapy. Understanding patients, and I'll talk a little bit about that later. When you develop a drug, how do you know which patients to test that drug in? It might be a drug that can help a lot of patients eventually, but originally you might want to test it on a subset to have the greatest chance of success, so you can bring that drug to the actual market to be able to sell it to everybody. And these are really important decisions. These are why natural history studies are so important. And this basis of our understanding here of this spectrum of epilepsy is important. If I put some of Praxis's programs into this space, you can see many of the programs on the left are programs that began in the lab, many in our lab, and are now on their pathway to being developed commercially, and they're the numbers of the internal programs, and I'll reveal what the genes are later in the talk. And then you can see that chunk in the middle is for the common epilepsies that affect many of the adult forms of epilepsy that are also a big burden for society that we're not going to talk about today, and I'll talk about the stuff on the left. But this spectrum allows us to populate our particular programs at various parts of the spectrum, understand how to develop it commercially, understand how to develop it scientifically and clinically. So it has been really important, and this is now the culmination, really, of about 80 years of research. It started in the 40s, it's now the 20s. It's been a long journey in our understanding to get to the point where we can have such a focused and sort of rational approach to developing therapies. Prior to this, anti-epileptic drugs were pretty much discovered by giving a drug to a model like a cat, zapping it with electricity and seeing whether the drug reduced its susceptibility to an electrical induced seizure. What they used to call phenotypic screening, and very blind to any mechanism. And most drugs stop the seizures, but don't do anything for root cause. And nowadays, rather than just trying to stop the seizure, it's very symptomatic relief. If you've got a bacterial infection, putting a Band-Aid on it isn't going to cure. You want to put an antibiotic on that kills the bacteria. It's the same thing with brain disorders. We don't want to just have symptomatic relief. We actually want to go for root cause, because the seizures should go away, but then all the other attendant comorbidities have got a chance of also reversing. And we're hoping to see, and that's so-called disease modification. We all know that the seizure burden is one important aspect of the lives of patients with rare diseases, but movement disorders, cognitive, ability to speak, all of these things are important in quality of life. And you have to have a different approach to how you think about treating these disorders if you want to affect that spectrum, that constellation of phenotypes that occur in the patients. There are two areas of, and this sort of gives a bigger overview of what Praxis is about, but on the left, it's that focus on rare disease where we have unequivocal evidence. It's a mutation in a SYNGAP, it's a mutation in an SCN2A, an 8A, and a PCH19. It's a specific mutation, and there's a specific approach developed for those types of mutation in that gene. On the right, I'll talk about that because there's also shared pathology. Often multiple mutations give rise to similar phenotypes, can give rise to shared pathology within the brain. There might be ways of developing drugs that have a broader effect across a common array. And we're also looking at those, and I'll touch on that with the molecules on the right as well. So I'll just give you a bit of an overview. And the whole point of today is not just to say, well, these are all the things that we're doing, but to sort of give you a flavour for what it takes to take an idea, a genetic discovery, that occurred 10 years ago, how to take that and to develop it and turn it into an actual drug that you can go to your doctor and get prescribed. And that's, the reason I've switched my career from purely research into development is to see that journey through in the sort of last stage of my career, to be able to try and convert some of those ideas into outcomes. And it's a whole new world. It's every bit as complicated, and unlike science, and this is not a criticism of science, in science many papers are wrong, and they'll be corrected. The literature will eventually find them, correct them, and that's just the way the scientific process works. In the drug development world, if a drug doesn't work, it doesn't work. You can't, there's no middle ground here. Either you develop something that works or it fails in a clinical trial, and that's the end of the story. So it's fairly black and white evidence. So you sort of have to get it right if you want to do this, and that puts more imperatives on it because it's huge amounts of investment from people. And it's this interface. You get investment from people who are interested in investing and making money, and that's their job, and that's okay. But then you've got companies who's passionate about changing the course of diseases, and you have to bear in mind that we have to make this work. Got to keep the investors happy. Have to try and deliver real products to develop our mission, which is to help patients and change the lives of patients. And it's balancing that is really important. So it's a very structured and disciplined approach that you need to take in order to move through that and optimize your activities to get the solution that you need. So the first thing I'll talk about is really this point on the right, this point around, I don't know if there's a cursor here, this will do. So the focus on these shared pathologies, and there's one program called Prax 562, and it emerged because this is a sort of a cartoon depiction of a neuron. And neurons or nerves that are in our brain, the unwanted activity of those is what epilepsy is. So these things, the neurons are sitting there, they normally deliver electrical pulses. The brain is an electrical organ. It communicates through electricity, and how it does the communication is through pulses, little spikes of electricity, bing, bing, bing. They're called action potentials. How fast they occur, how they're grouped together, the timing of them, how one neuron communicates to another through that network. They're all connected into these networks, talking to each other through these electrical spikes. How they do that is critical. Normal behaviour, it lets me speak and stand and function. But if it goes awry, it causes epilepsy. There's too much unwanted electrical activity and the wrong type of patterns, you get seizures. Now, there's particular parts of the neuron. There's this area called the AIS. It's called the axon initial segment. It doesn't really matter what it's called. But it is the spot in the neuron that actually generates these electrical pulses. There's one part of the neuron called the dendrite, which is the Latin word for like a dendritic arbor. It's part of a tree. And it's the receiving structure of the neuron. It's out there listening to all the other neurons and all the chatter. All that information comes in and a decision is made at the axon initial segment as to, okay, I've heard all this stuff. What am I going to do? Well, I'm going to fire off my own electrical activity and contribute. You can imagine that if you listen to a feeding hour at the zoo and you hear the monkeys talking to one another and you hear the chattering, they're all communicating. Even though it sounds chaotic, one monkey will do this, another will chatter, and they're all telling each other that the food's coming and will change your behaviour. Well, neurons do the same thing. They're listening and then they're participating in the chorus of brain activity. Now, this part of the initial segment is where that decision is made on how am I going to respond, what am I going to say based on what I've heard. We know that many, many important epilepsy genes are there. 1.1 is the gene that causes Dravet. 1.2 causes autism, causes epilepsy. 1.6 a major encephalopathy gene. So really important genes are at this point, and we know that when this part of the gene goes wrong, seizures can go awry. So Praxis, based on this understanding, wanted to develop a drug that helped tame the function of this part of the nerve. And I won't go into all the gory details. These sodium channels are little electrical switches, act like little taps, and they let a little spurt of water in. So you can imagine if you had a tap that was, I don't know if you remember your primary school, you would turn the tap, let go, it would always spring back so you couldn't leave it on. Sodium channels like that, you turn it, and a little bit of water that goes through, you let go, and then it shuts. Now, that's how a sodium channel works, and that's how the basis of electrical signaling. That little squirt of sodium going into a cell, into the neuron, sets off an electrical cascade. Now, if that tap doesn't turn off properly, all havoc starts to wreak. And it's called persistent current in the biophysics world, but it's a dripping tap in our world. And if you can block that drip, but still let the tap work, but block the drip, you can have a big effect on the way the electrical signaling works. So Praxis developed molecules. You see that. This is in a SCN8A patient. You can see, rather than in a normal patient, you can see the black line. This is the sodium channel opening, the tap opening, and then snapping shut. This happens in 1,000th of a second or less, that little blip. And it goes back to zero. But you can see in the patients, patient 1, patient 2, patient 3, it doesn't go back to zero. It's a tiny fraction. It can be as little as 1 or 2% of the total amount of the tap being open, but it's enough to cause the disorder. So if you can, and it also happens in 2A. This is a 2A patient. Normal above, 2A patient in pink, you can see that it doesn't go back to zero. So the idea is, can you make a drug that specifically targets that molecular process? Then you might be, and it'll work on multiple sodium channels, whether it's 2A or 8A, it'll work and it'll interact and be beneficial, broader than just one particular genetic disorder. And the idea is, so for a nerve, you want to be able to spare normal activity. So we look at this thing called an input-output curve. I don't know why that, is there a way to get rid of that? The thing at the bottom?
There you go.
Oh, okay, good. I won't touch the screen again. The input-output curve, basically, when a nerve works, as I said, it receives input and then it needs to do something. So normal functioning requires the response to a certain input. So I hear something, I say the appropriate thing. Danielle says, "Hey, how's it going, Steve?" I say, "Oh, terrible." And so that's my standard response. But if all of a sudden she asks me a question and I just, explode and start talking randomly in random words, well, that's what epilepsy is. It's that inappropriate response to a normal stimulation. And you want a drug that lets the normal activity, so on the left part of that curve, to work unabated. So when the neuron is not being overly stimulated, it can respond and it can control the things we want. Speech, movement, cognition, all of those things. But then, if the nerve goes into this activity where it's really uncontrolled, you want the drug to really be effective when it's doing that, but not effective when it's doing normal things. And that's why drugs have side effects. Because you give a drug that just dampens down everything indiscriminately, sure, you'll stop the seizures, but you'll also sedate the patient and start to have other effects, in which we see with a lot of anti-epileptic medications. They're indiscriminate in the way they act. Can we put some discrimination in the action of the drug? So and I won't go into the details of the biophysics, it's not really important, because in principle, it's can you achieve that ability? And that's what we're working to achieve with these molecules. We think we've got there this small molecule called 562 that's a sodium channel blocker that does that, and we're going to target it against SCN8A, SCN2A, and even some patients with tuberous sclerosis, because we think that part of the gene goes awry in tuberous sclerosis, and we know that there are patients with TSC1, TSC2 mutations that cause seizures, and these little malformations in the brain. And we know surgically, if you can remove some of these tumours, you can stop the seizures as well. So there's something about the neurons that are within those malformations that are very active electrically And I know there's some really great groups, and one in Melbourne, Simon Harvey and Virginia Maxima that I've been working with a little, that are trying to think surgically what's the best strategy, but is there a chemical strategy we can do to support those surgical efforts as well? This sort of drug might be really useful in that way. If you look at overall, because if you include TSC and you include 8A and 2A and maybe other maybe this might work in KCNQ2 as well. You can see that there might be 10,000 or more patients. And it gives a good market size, so it lets you justify the effort and the expense you need to move into these spaces, but it's got the chance of helping lots and lots of smaller groups. In the same way, Daniel talked about GETA being the aggregation of lots of smaller things becoming a big thing. Well, our approach is sort of a parallel to that. And I won't bore you. These are the normal drugs. Lamotrigine, carbamazepine, you've probably heard of those, and I'm sure many of you have had these prescribed. But if you look at that, these lines, so you look at the blue line of the praxis, you look at the persistent current block, look at the dashed black line. You can see on the lamotrigine and carbamazepine, 10 micromole, 100 micromole before it's effective. The praxis one is about 10,000 times more potent against that persistent current. So that's the thing, what we call is differentiation. How does this molecule differ from the things that are already out there? And can that difference be beneficial clinically? We've got a scientific hypothesis, we've got a good biophysics, animal modelling we can show here, and you can see here this huge left shift in Prax 562 relative to these other molecules. It's more effective against that type of sodium channel activity than the normal type. It's got a 60-fold ratio of persistent to peak inhibition, that's 60 times more effective against that dripping than against the main current. So we hope that it can block pathological activity and spare normal activity. When you give it to an animal model, you can see on the left, there's a seizure model, and as you increase the amount of drug, you can have a big effect on the latency of these seizures. And you can see, so we can get a half activity there. We can say at a certain dose, 2 milligrams per kilogram, this 50% of the effect is seen. On the right, you can say, well, if you give this drug, does it have an adverse effect? Does it stop the animal's ability to move? And SLMA is just a locomotor activity. It's a standard way of testing drugs. You give it the drug, the animal starts to get sluggish, and you can actually quantify that. That ratio of the dose that's required to cause the adverse effect, the dose that's there to cause the efficacy, gives you a therapeutic window. Here, for Praxis, we got 17.2x. So that means it's fairly broad range, so you can be effective without starting to go into the side effect range. And that's what we're hoping to achieve by targeting specific biophysical properties. When we give this drug to a 2A and an 8A model, mouse model, very efficacious. You can see 100% block of seizures. On the left for the 2A model, on the right, 8A model, 100% block of seizures in the 8A model when we give this drug. So really effective against two of the genetic disorders we want to target. All this is really important when you're thinking about delivering a drug into the clinic. If you can show, yep, does it work in the animal models that have the same mutation? Yes, tick. That's a really good starting point. And we keep accumulating these small series of ticks, and it gets more and more compelling around why this drug might be useful in people. And we have another model that's the similar sort of drug in looking at survival now in 2A and 8A animals. And you can see on the left, untreated, the black line, all the animals are dead by 80 days of age because of a really bad seizure. You treat the animals that are treated, the yellow line above, only a very small fraction of the animals died whilst the drug was being onloaded because they start dying so young. Once the drug was on board, you can see they were completely protected. They weren't having seizures, they weren't dying. Same for the SCN8A, and when you took the drug off, seizures came back and the animals died. So this is really good preclinical evidence that we're having the effect. We're affecting the seizure model. We're not having a massive effect on movement. These genetic models with the actual changes, we're seeing very specific and encouraging changes. And it even works in a KCNQ2 model. So we tested that because of the KCNQ2 gene is also in that initial segment. So we see anatomical and biophysical and neurophysiological convergence, and maybe we can see some effect of that coming out in the different phenotypes that it can treat. So that was that idea that can we, by globally understanding pathology, can we develop a drug that's broader and affect a broad class of epilepsies? That's something, and I'll talk a little bit about the clinical trials in that area later in the talk. Similarly, for other genes, and you can see here, PRAX222. This is something I began in 2015 with two parents that were from Alex Nemiroff and Kelly Delby. They both were two parents, had two children from obviously from different families, but they were passionate about trying to do something to deliver a medication. We started a small company called Rogcon. We're collaborating with a big American company at the time called Ionis, and we developed an ASO to try and treat the gain of function cases for SCN2A, and I've been working on that for many, many, many years. I'm just delighted to see it's gone from a lab idea into a thing that we're developing into a drug. We're on the verge of trying to start clinical trials now with this molecule. So it's in the US, there's about 1,500 patients. They're not huge numbers. For the gain of function cases, we know many cases here in Australia as well. We know of cases in Europe. I know that Chris Pearce has been a major advocate now globally, really her reach and her impact. She's been talking with Praxis and all the other organisations around trying to move the needle for 2A, while she's working tirelessly for GETTR and for everyone's benefit in other cases as well. We're really interested in this. I'll show some data from that. SCN2A, I said it's in the initial segment, that hotspot of the neuron. Now, this gene does too much in these cases. For these kids, it's overworked, it's overly active. And the idea is, can you remove some of it? If it's too much of a good thing, basically, can we use this mode called antisense oligos to actually reduce the amount of SCN2A? That's what we've taken our time to develop, an antisense oligo that selectively can reduce the amount of this channel that's overactive. You can see on the left, as we increase the amount of drug, we go from no effect to a massive effect. And so we can, depending on the amount, we can go leave all the channels there, we can get rid of all of them with a high dose. The idea is to find what's that sweet spot. How much reduction do you need to do, and how do you deliver that clinically is the clinical challenge, and I'll talk a bit about that later. So this is just on the right is just another confirmation of how we look at that. This is the dose in the animal models. This is survival over time. So we've made a genetic model that has exactly the same mutation that the patient did, that had these seizures. Untreated, these animals die after 20 days. If, as you can see in the blue and the yellow, is a low dose and a higher dose, and you can see as you treat them with more and more drug, you have more and more benefit. So you can have a major effect on the life of these animals. They stop having seizures, and if you measure their behaviour of the animals that survive, I can't tell them apart from a normal animal. So it's not that they're just not having seizures, all the other behaviours that we measure are identical to controls. And that's such a great sign. So, by no means is it, are we certain this will happen in people, but it's really encouraging that in a rodent model, you're not just affecting the seizure axis, you're affecting their cognition, their movement, their social interactions, all these other functions that are important to make a mouse, and presumably the same thing to make a healthy and happy, high quality of life individual. This is a different dosing paradigm where we gave a second dose. You can see how much more effective it was as we kept dosing these animals. On the left was a single dose when the animal was 1 day old. Now, if we gave a second dose when they were 30 days old, antisense oligos have got a long life. When you give them in the body, they hang around for 2 or 3 months before you need to give and you've probably heard about spinal muscular atrophy. There's a drug called Spinraza that's having amazing effects on children around the world, including in Australia. Trials run across Australia for that molecule. Same sort of medication here. When we give to an older animal, we still see benefit. And this is an important experiment to say, because you're not always going to treat a kid at or around birth. They're going to be older by the time we start to treat. Is there a chance that we can have an effect? Yes, there is. These are the animals that are on the precipice of dying. 15 days of age, at 20 they're all dead, rejected at 15, you can see the flat line, those animals survived that treatment. Hope that we can go and have an impact later in life. Trying to define that window of how wide a window it is before you can have, the effect is less and less marked. And I hope that effect will be as big as possible, that no matter what age you are, you receive some benefit. Because that gene is overactive all the time, even in a kid who's 20 or 30 or an adult who's 40 years of age. So having an effect on the gene and calming that down, we hope that there's going to be some benefit from that, even though there might be some long-term effect of living with that gene for a long time. We always hope that there will be some other benefit of dealing with that gene's mutation. Praxis 20. There's another mutation in a gene called KCNT1. We know of cases in Australia. It's something we've been studying a lot in the lab as well. Rather than a sodium channel that lets sodium in and out of the cell, this is a so-called potassium channel that lets potassium in and out of a cell. And really, the electrical signaling in the brain is fundamentally caused by the balance of these two ions, sodium and potassium, how they're situated inside and outside of a cell, how they move, determines the electrical state of that neuron. This is a mutation in these kids that's gain of function, so too much KCNT1 activity, and these kids have hundreds, some of these kids are having hundreds of seizures a day. They don't move, they don't talk, they don't walk, they sit in a cot, they have lots of seizures, and many of these kids die very young, from 4 years to 10 years of age. Really terrible prognosis, and caused by what seems like a very easy to define genetic change. Genetically, we can detect it. Functionally, a week in the lab and we can confirm that it's gain of function. So we want to think, how do we go about solving it? We've looked at what's called a small molecule program. So rather than an ASO, it's a drug. And you can see here, Praxis developed a blocker of that channel. So a little small molecule, a pill that you would take, and it would stop that channel from working. Here, we've made a whole bunch of common mutations that you see in people, and we've given the drug, then you can see that as you give more drug, you can get 100% shutdown of that channel. And so we know that it doesn't just work against the wild type, but it works against most of the mutations that we can find, which is a great sign. This is an animal model of a human mutation. It has seizures, it has electrical discharges on the EEG that we can detect. You can see on the left, as we dose up, you can see the grey bar, that's the normal level of seizures. As we give higher dose, we have essentially about 100% effect, 95%, almost stop those seizures cold by dosing with this drug. We also see, as I said, on the EEG, there's abnormal electrical activity. That's the grey. You can see this is a relative amount of spiking. And as we dose with the blue and the yellow, low dose, high dose, we can have a massive effect on that electrical discharging and the EEG. So we're having two really important effects on the electrical activity of the brain. We're reducing or completely stopping seizures, and we're restoring EEG function. Back on, David. We're all good? Okay, we're back. All right. All right, sorry. So let's get back on. So Praxis 90 is a program that's designed to, therapeutic program for SYNGAP1. SYNGAP1 is, and you can see the number of patients here in the US, some of the symptoms, and what the treatment goals are for the program. Good number of patients in America that we can justify this commercially. 1,600, we think it's enough to be able to engage, make a drug, deliver it. And also there's going to be patients in Australia, as we know about, and globally. SYNGAP is and I mentioned before in those gene classes, what I've been talking about before is that top left thing, which is ion channels. On the right was proteins in synaptic support. So ion channels are the proteins that are important in how the neuron signals, but how a neuron talks from one to the other is through a synapse. So it's the mouth and the ear. So one neuron's got the mouth and it's talking, the other side of the synapse is the ear and it's listening. These proteins that interfere with those two functions can have a big effect on patients, obviously. SYNGAP is intimately involved in that process, and it's a loss of function. There's not enough SYNGAP to support function when you have a mutation. So the idea is, can we get our ASOs are good drugs in that regard, in that we can use them to increase the amount of the gene as well. Not only can we reduce a gene in one mode, but we chemically alter the ASOs and we can make them turn a gene on. So can we use an antisense oligo to increase the expression? This program really began as a collaboration between myself and Danny and Danielle, probably about 4 or 5 years ago. 6 years ago, oh my God. 6 years ago, where we came together, we were interested in working on it. They helped us get some really key, so they helped us get some funding that ended up coming through the Epilepsy Foundation via some lobbying that they did. And that helped us get started on this such that I could build a program that would attract commercial support. And that's really important. So having just a raw idea on a piece of paper is a lot harder to move that program through because there's zero proof of concept. But that money enabled us to say, look, we can actually make some test molecules that have the right effect on the gene. They can increase it. We've identified parts of the gene that are important for this process. So we can put all those things together and we can go from several $100,000 to the tens of millions of dollars that are going to be needed to make this commercial. So it's been a great outcome of show. And the same way RogCon with the 222 happened in the same way. Parents getting involved, helping us deliver on important research goals that then cause further investment in a commercial level. So SYNGAP1 is now on that path. One really important aspect of this, and there's been a little bit of controversy in literature, is, and it goes back again to that question David asked, well, can it be too late? If you turn SYNGAP on in a kid that's 5 years old, 10 years old, is it too late? Or 1 year old? We don't know. And there was an early paper by the same group, by Gavin Rumbaugh, who sort of said, well, you turn it on, doesn't help. So maybe it's not worth, it's going to be a very difficult therapeutic decision to make. He went on and did a follow-up study, a little bit more detailed study, and showed that if you turn the gene on later in life, in an animal that didn't have it before, that you could have restoration of EEG function, of seizures reduction, memory improvement later in life. Again, back to that thing that I showed with KCNT1 blocking. Later in life, you can get benefits. And I think it's great to have the preclinical support of that. We use that to motivate ourselves internally. We use that in the story we tell to investors because all the evidence lining up to say, well, not only is this going to be a great drug for newly diagnosed kids, we think there's window here to help all patients. And the more evidence we can have, so I've been collaborating with Gavin now for a while, and we hope to have even stronger and richer collaborations with his group at Scripps in Florida. And now this just shows work we've done in collaboration with Praxis. So Praxis has got a collaboration with Florey Institute where they've entered into a funded research collaboration. We've produced an antisense oligo that can have a 2.5 to 3 times increase in the message. So the expression level, genes go from, the gene is the major plans, the RNA is the blueprint on which you build the house, and the house is the protein. This is the blueprint, more RNA. So we're having a almost threefold increase in that level RNA, which is exactly what we wanted to see. So we've now made ASOs that can increase it. We now have to test, does it work it works in the dish? Does it work in a whole organism? Is it potent enough? What do we see? Is it tolerated? But we're on our way here, hoping to deliver another candidate next year for SYNGAP1, and to try and see whether these sorts of ASOs, what's it going to take, if we can get a safe and effective molecule, what's it going to take to find the patients, etc. I'll talk a little bit about that later around the next steps. Once you've gone through all this work in the lab, and you've produced molecules, you then go through this process called GLP, which is it's called good laboratory practice. It's a standard, a compliance, an auditing standard that says you've done the safety testing in a way that meets future human use. And they're very, very cautious, whether it's the TGA in Australia or the FDA in the USA. They've got quite stringent criteria to show that you've done all the necessary things with all the right quality and demonstrable quality and demonstrable paperwork, that this thing is going to be safe, because first and foremost, safety is always the first hurdle for a new drug into patients. And we're going through, that's going to be the next process, and then we start to think about dosing in people. Talked about those, so I'm going to. So I think this is we're now at the point, if we go back, I'll talk about these two programs that we hope now to develop, and talk a little bit about the clinical development. So PRAX-2.2 has gone through that safety testing. It's now ready to be put into patients. And I'll talk a little bit about what's required for that, and similarly for that small molecule 562 that we hope to work in a lot of different DEEs. What's required of that? And this is a part, really, another area where families, patients, patient organisations can really help, not just practice, any company that's working in this space. Clearly, we've got our preclinical case that shows that its potential to be disease modifying. It affects the mice, good survival. When they're treated, these animals look like normal mice for all intents and purposes. We see effects on seizures, on interictal discharges, all the behavioural, and this repeated dosing causing continual survival benefits. And the idea is to make an antisense oligo that we administer through lumbar puncture, through a spinal tap, and that's the standard way that these antisense oligos are injected. You inject them through a lumbar puncture, it goes up and it circulates around the brain, and you need, hopefully, you look for a drug that's stable enough that you do this injection only 3 or 4 times a year. And that's the same thing as Spinraza. So that's the thing, we've got to make a molecule that's stable, that's safe, that's long-lived enough, and has the effect that we need. And importantly, we know how much to give. Dosing is a big part, as you'll see. How much to give of any drug is a big part of what you think about when you're doing clinical trial delivery. Now, you talked about the natural history studies. This is a work that Praxis has done with a collaborator, looking at historical medical records in patients with 2A mutations. Normally, natural history studies are prospective. So you start today and you recruit patients into that study, and then you look for the next 3 to 5 to 6 years and ask the question, what is an individual patient's journey through that disorder? What happens when they're diagnosed, when they're 1-year-old, 2-year-old? What happens with another patient? Are they similar? Are they different? If you've got exactly the same mutation, do you present the same? Lots of questions you can answer about how that disease unfolds individually and collectively. And then that's important for saying, if you're going to test a drug, what are you going to test it against? Seizures? Development? Movement? Speech? What thing do you, and you don't want to pick them all. You don't want to make the hurdle so high, because you have to nominate. We're going to give this drug at this level, and we're going to impact this thing about the patient. You get that wrong, program will fail and it's gone. It's not just, it's failed, let's try again. It can be gone completely. So it's a huge investment, tens of millions of dollars into that. So how do you know what to measure? Seizures are always good. They may not be they may not be the most important thing. They are an important thing, but maybe they're the most tractable thing. And we didn't even understand in 2A how many seizures were the kids really having. A lot of these kids are being treated with various anti-epileptic drugs, phenytoin included. And so you don't, how many seizures do you see when they're on medication? You can't take them off the medication to do the trial because there's ethical concerns. You don't want to ever make the kids worse. So you're saying, well, if they're not having seizures, but this retrospective, this was by looking, and in America, we've got to admit their electronic medical records are phenomenal because they're centralized. So there's the ability to be able to follow patients, and there's 10 years of data, 20 years of data in some of these kids. You know, obviously the diagnosis wasn't made until the kid was 15 years old, but then you can look at the medical records for the 15 years prior to that. And when you look at this, we've got a nice idea now of how many seizures these patients have been having. And then you can say, well, if we're going to use seizures as what we call so-called endpoint for a trial, if we decide to do that, well, we know that you have to look for 3 months or 6 months to be able to do a proper before and after comparison. You can't look for such a short period. Well, they only had one seizure in that period, we gave the drug, they have none, isn't very compelling evidence. But if you look for 6 months and they had 10 seizures, you give the drug, you look for the next 6 months they had one or none, well, that's pretty compelling evidence. So how long should that period be? What feature? All interested in development. These are neurodevelopmental disorders. But if it takes 3 or 4 or 5 years for the development to be delayed, how long does it take to be restored, even if you have the perfect drug? I don't think you give a drug, then all of a sudden the child will start speaking. I think it's like any physiotherapy, any of these therapies, you break a leg or whatever, it takes 6 months before you can restore motor function. You relearn, you've got to retrain your brain. Even if the brain now is normal, for all intents and purposes, you still have to retrain it to recapture what was lost. So how do we do that in a clinical trial? Can we do that feasibly? Has anyone ever done that before to give us comfort that we might even be able to do that? These are the sorts of questions that we ask clinically. The more data we've got from kids that are treated, 5 minutes, okay, I'm almost done. The more data we got, all of these things are really helpful. That's where patient families are really important. This is what we did for SCN2A. Talked about the other things we measure, number of medications, all the things that have happened. You look at some of these things, diagnostic and therapeutic procedures for 2A, up to 40, average of 11 hospitalizations, 26 different medications, 99 hospitalization days on average. And then they're massive numbers considering, you know, most kids never go to a hospital in that period. These are big numbers. So we can understand the burden. We've got an EEG signature that might help us. Kids that are having these seizures have a higher burden of intellectual discharges. So the EEG is not normal, even between the seizures. There's something different about that. Could we use that as a marker, etc.? And these are the principles that we want to follow. Because they're small populations, normally you do phase 1, which is just safety. And for a normal small drug, you do that in healthy volunteers. You know, generally, depending what age you're doing it, normally it's for you know, college kids wanting to make a few extra bucks, go into trials and take these drugs. We see that they're safe, and then we start to give them. But with ASO, you can't do that. So we want to, you know, can we design a trial where the patients, we do low dosing for safety, slowly go up, see how they're doing, but then they get enrolled in actually this phase 2, which is looking at some sort of efficacy, etc. So we consider all these different things. What's the right placebo? Are there kids that should not be getting the drug? What are the ethical things? What are the strengths and disadvantages of doing that? What patients, of all the patients that are available to us, which ones are the best suited for this? You may pick some that we think, well, these are the ones that are going to show the best signal to best demonstrate efficacy of our drug, and we believe there's going to be a global benefit, but this is going to be the way of really fast tracking and showing that we get the efficacy we need to get the drug into the broader population. How do we analyse the data statistically? What are the endpoints? And I talked about that. Seizures, neurodevelopment, cognition. How much do we give? And how do we get there? For SCN2A, you might be able to overdose. Some drugs you can't, some drugs you can. How do you approach that Goldilocks spot of treating without overdoing it? All these things we factor in the thinking about how to engage into a trial. Again, data from, you know, the more data that's aggregated globally about these patients, if family groups get together and share data, do all that, it's really, really important. Natural history studies are really important for that reason. I'll leave it there for now, giving you a taste for how we go from genetic discovery and a journey that began in the 40s to our ability to deliver precision medicines today. And some of the challenges, not only there's the initial clinical and genetic studies, lab studies to understand how the gene actually breaks the brain and the mechanisms and ideas for therapy, now to how you actually take all that and turn it into a new medication. And I'm glad to say that there's not just Praxis, but other companies are starting to do this. So we hope that for every single gene mutation, there will be some therapy we can attach to it. And I'll leave it there. Thanks very much.
Maybe, do you want to use that? And I'll
Sure.
use the podium.
Okay.
So you can move around a bit. Thank you very much, Steve, for that. So the questions from the room that people would like to ask? Jenny?
Hi, Steve. So, I feel like, as you touched on when you were talking about endpoints from our family's perspectives.
Sorry, I think they want you on the microphone so
Yeah,
that
sure.
Zoom participants can hear you. I'll just speak here, David.
Yeah. Great.
So from a parental perspective, when we've got a kid with one of these conditions, obviously you've discussed a little bit on endpoints. There's seizure activity on the one hand, and then concerns with their development on the other hand. And for like a condition like KCNQ2, a lot of, like Ingrid was in her speech last year explained that there was not a big correlation between seizure burden and developmental impairment, in that you could have a lot of the kids will be fairly well controlled from a seizure perspective, but severely developmentally impaired. So you mentioned a couple of years ago, like about ezogabine. I'm muted.
The batteries are out.
Sorry, Jenny, just keep
going, I think, yeah.
Okay, so check now. So obviously ezogabine is like the closest thing we've got for a targeted gene therapy for KCNQ2 because it affects the potassium channels, but it was obviously grandfathered because of concerns of retinal pigmentation. It's now being brought in via the EPIC trial, but the EPIC trial, again, like you said, is limited to kids that are still having active seizures because you don't want to make them worse. So my question would be, when you were talking about PRAX 562, because the kids have got well-controlled seizures, we can't get the ezogabine, and then the ethical question is, would there be a difference enough in their developmental input? Would there be a possible developmental improvement that would offset the risk of some retinal pigmentation side effect when their seizures are well controlled? And that's one question. So
let's break. Can we
Okay,
do that?
I'll do that question. So the question was, if you have drugs that are affecting only the seizure domain and not necessarily targeted towards neurodevelopmental outcomes because of the mechanism of the drug, is that still a good thing? And is the risk of, I guess, retinal risk, that's an ezogabine specific risk. 562, of course, is not an ezogabine drug, so it'll have a different risk profile. It shares, so the concept there is that it shares very fundamental biophysical overlap with KCNQ2 and 1.2. So that initial segment is also full of KCNQ2 channels. So we think that the part of the neuron that's being affected by 562 may have more overlap than just a general drug that increases GABA and turns the brain down, for instance, and has some effect on seizures. So there is a chance, so you're asking, is there a chance for disease modification with these other medications?
I suppose the other part of the question with PRAX 562 was that you were saying that you needed a lower dose. So a question a lot of parents ask themselves is, well, I'm sure that all of us in this room, you got your kids on these fairly heavy anti-epileptic medications, and the question is, if I took them off the medications, would their development be better, but their seizure risk be higher? And then you don't know, and the specialists don't seem to know. There's figures like, maybe 2 years seizure-free, and then you try to wean it. There's stories, certainly I only know the KCNQ2 community, where, you wean off the sodium channel blocker, and 5 years later, suddenly they start having seizures again and they've got to restart on medications. That's obviously like in the back of our mind. So you don't know whether or not it's like, what's the drug side effect on their development? Like, is it possible that PRAX 562 would have because it has a lower threshold that you could use lower doses to get the same seizure control, but then because you've got a lesser drug burden, you might get an improved developmental profile.
That's always the hope. Because if you think about it a slightly different way, that a drug that's more proximal to the root cause, I think is going to have a better chance of not only affecting the seizures, but having an effect on everything else. We can argue that 562 is more proximal to the genetic lesion than a GABA drug that's way downstream. All this stuff has happened and then a seizure occurs and we're stopping the cortex from having a seizure. That's when you potentially run into these problems. Okay, we can block seizures, but we're not stopping all this other stuff that's happening that's potentially impacting development. And we try to become proximal either through genetic mechanism or through shared pathology, but getting closer to the root cause in each case. And then we're only going to know through clinical work in the end. You're a physician, you understand that. We'll do we do a phase 2 trial that shows some efficacy? We do a phase 3 to register. But then there's going to be trials done, phase 4 or investigator trial that'll start to look at the 5-year and 10-year outcomes that are going to occur. But I keep going back to that idea, the more proximal you are to the genetic lesion, the better chance that you will have multiple things under control with fewer burdens.
Which is
Great.
obviously
Thanks,
a huge.
Can we
Could I just
give
make
others the opportunity?
Just one quickly thing. Will that become available to people whose seizures are well controlled? Because they're not going to be allowed to go onto the trials because if their seizures are well controlled.
Well, we don't have the criteria published yet. We don't know. We're still thinking through all those things, Jenny, about exactly who can be in, who can be out. And dealing with all the ethical issues around each of those as well. It's still a work in progress.
Great. So while Danny's getting to the next question, you got let off the hook. What's your first thing, Steve? Danielle gave you the heads up. What's the thing you're going to do for the first time?
For this year?
Yeah.
Doesn't have to be related to science.
No.
So this is a bit embarrassing.
Mine was about a cow.
It's a bit embarrassing, but my first thing is that I've got a million hobbies I've started and never done, just never have the time. So this year I got fascinated by the IBM golf ball typewriter. I'd never ever seen those. They first came out in the 60s, and it's one of the most elegant mechanical machines. It actually is a mechanical machine that works digitally. So it's actually a little computer that uses a mechanical version of digital encoding to turn that golf ball around and type it. So I'm now a typewriter repairman. So there you go. That's new for this year.
Nice.
It's a bit pathetic, but new.
Nice.
And my wife now wants to leave the room.
Sorry, question from me. My daughter has SCN2A. Initially, our biggest issue was seizures. Now she's 6. We've moved into seizures and movement disorder. The movement disorder is probably the hardest part now for us. In your mice models, have you been able to replicate any of the movement disorders seen in SCN2A?
Yeah, can I ask how old was your daughter when she presented with the first seizures?
She's gain of function, so
She's
she
gain of
was,
function.
day 1.
Yeah, okay. So so we see that, often those movement disorders and the chorioathetosis is seen more pronounced in the loss of function cases, paradoxically. But we have seen some 2A, the gene itself, is an important part of the brain that controls movement, called the cerebellum. And again, we saw, obviously, these animals, when they were untreated, they were young, so they're only 20, they were just coming off milk. And they would have terrible, where they'd walk around the cage, they'd be vibrating and have terrible tremors. So when they were treated, some of that stuff was reduced. But we didn't specifically run a movement because they were so little. Normally, the animals need to be 30 days plus to do more pronounced. But when we put them on you can put them on a machine that measures the properties of their stride, it looked like a normal animal. So the stride, the properties of their striding, you can measure when each foot hits the ground and the coordination, because a 4-legged animal has this beautiful coordinated, and you can see that, and you can see the swing time and the gait, all those things look normal, but we didn't have animals that were 30, 40 days of age alive to see what they would have looked like without drug. So, but it speaks to your question a bit, and then we hope, again, because we're fixing the root cause, we hope these other things don't emerge, the movement disorders and the other things, but I can't provide strong evidence for gain of function now, because these animals, no matter who makes them, always they die really young, because they have such severe seizures.
Other questions from the room? Thanks, Danny. Yeah, and so people that are on the live stream, feel free to ask questions via the chat function in the live stream, just the little chat box top right-hand corner. All right, thanks.
Thank you. We have a daughter with a fairly recent diagnosis of ATP6VOC, which we understand is is a protein coding gene. You spoke a lot about sodium and potassium. I'm not sure if I'm articulating this correctly. ATP6.
1A3 or no?
ATP6VOC.
Okay.
Don't know that one?
No, I'm not
I
familiar.
just want, there was a lot of discussion, I suppose, about sodium and potassium. I was just wondering if there was any research being done in the protein coding area.
So, yeah, in terms of a gene makes a protein. So all the things we look at, we do look at the proteins. Now, we haven't looked specifically at that protein. I'm not that familiar with it, to be honest. But we always look at the proteins that are made. So the gene is the plans for a protein. We make an assay that lets us study how that protein works in a cell or an animal. But specifically for that gene, I have to look it up. I just wasn't, I know the ATP 1A3 and others that are sodium pumps, but I'll have to look that gene up. It sounds relatively new.
It is, yeah.
VOC6.
Yes.
Okay, there you are, stumped me.
Don't worry. Thank you.
I'll hold it. So.
Yes. Is Praxis doing any work on SCN1A?
So I started when we sort of got Praxis going, one of the ideas that started it was we discovered with collaborators in Queensland a spider venom that worked on SCN1A, and we published a nice paper showing how we could fix the mice, the Dravet mice, by treating with this. The difficulty is it's a peptide. So spider venoms are little peptides that are really hard to deliver. You can't deliver it you can't take a pill, you can't take an injection, you have to deliver it directly into the brain, and it gets destroyed by the brain CSF within hours. So you have to continue. So that became a really difficult task. So I'm always interested in looking at a small molecule that does that. I can't say anything specifically now, but there are numerous companies working on Dravet, Stoke Therapeutics, Opco. Yeah, Encoded. Lots of companies looking at it. There are companies looking at delivering a whole new gene. So there's a lot of activity. Zogenix had a drug called fenfluramine, and that was recently acquired by UCB, I believe. So there's a lot of activity, a lot of options for Dravet, and so it's getting a little bit of a crowded space. But I haven't given up thinking about it, and it is an important gene as well. 1A could be an important gene for all epilepsy, not just Dravet.
Thanks very much. So we have one last question in the middle at the back, Danny.
Thank you. Hi. Oh, that's better. So I guess when we were first diagnosed with STX-BP1, we were the only family we knew at the hospital. Now I'm excited that we're sitting here with 4 families from Australia. Seeing the graph that you had that showed the conditions grouped together, obviously the one that stood out to us was SYNGAP because it shares a similar, I guess it's a cousin condition. How as a group of families do we then lean into that and have a research done on our particular condition happening simultaneously, piggybacking off similar conditions, where the function,
Overlaps.
Overlaps.
I think what's interesting, if you look at those 4 boxes I presented, it almost represents 4 separable scientific disciplines. There are groups that focus on synaptic function, there are groups that focus on ion channels, there are groups that focus on structural abnormalities, and those that function on gene regulation. So you could think as STX-BP1, I think it's Monk 19 as well or something. So it's been studied a lot for synapse function. Maybe there are groups that you might support from a scientific perspective and do fundraising, whatever, that can collectively help everybody in that area. Because there might be, again, shared pathologies and sort of points of convergence of all those disorders where one approach might help many. And it's more likely to occur if you're in the same physical spot and if you're at the synapse. So that's maybe one way you can lean in. I still suggest we all lean in collectively, because this collective is really strong in looking at DEEs as a whole and looking at all the subtle differences. It's still really important, but then if you specifically lean in and support the research in synaptic function, and there are great groups all over the place, at the Florey, up in Queensland, Fred Meniere is one group that's a really expert on that same protein you're working on, are interested in, and those groups could benefit as well.
Now, we've had one question from the live stream.
Yeah, sure.
On, I think this is on. Steve, the live stream question is asked, are there any updates on DNM1?
I know that people are looking at, companies that I can't disclose are interested in DNM1, and the approach there. DNM1 there are some common variants in DNM1, and again, that's a dominant effect. So there could be an ASO approach there that companies are working on. And I know there's one company called Dynacure that worked on a related one, and that's the sort of company that might be interested in DNM1. And you might want to reach out to Dynacure and see what their interest might be in DNM1.
Thank you very much.
All right, thanks, David.
Thanks a lot, Steve.